Blue. Okay. Good.
All right. I was hoping a little bit more people would show up. No, they might trickle in though. Just for a couple minutes. Wait it out. So considering I have invited almost 100 people to this review, this doesn't surprise me. This is how these reviews go because they're optional. Most people don't show up or they're working or something like that, and I understand. I can accommodate for that. This is security. There is no advanced. Maybe they changed things when they updated it to like Zoom Workplace or whatever this is. I'm trying to find a way to like have more than one. I don't know if you guys know this, but the way the Zoom works, you can log in on one device. It's like your computer or your tablet or your phone or whatever, but you can't log in your same login identity with more than one thing. If I could do that, then I could log in with my tablet here and log in with my computer here, and then start like basically watching what is on my tablet for you guys to see, so I could actually write on it with like a pen and do all this stuff and whatever. But they changed it whenever they updated it to Zoom Workplace, so I'll figure it out eventually, and it'll be better, I promise. But for now, you get to put up with me if I write on this, and I probably won't because I want to keep this short. But if I write on it, you're going to get to see me writing with my cursor. So sorry, that never looks good. So we're going to go over week one. We'll go ahead and get started. And we are recording, so you can always refer back to this if you need to leave or whatever. We're going to go over week one. And that means on Monday or Tuesday, we went over chapters 1 and 3, and then on Wednesday and Thursday, we went over chapter 2. So we're just going to briefly run down the topics that you should have, gleaned the general idea of briefly, and I'm not going to go over them or review those. I'm just going to remind you that that's what you should have learned. The things we are going to go over are the topics. The only thing I'm really going to spend time on is the topics that people miss on the exams. So these are topics that people got, it was like 50% or less of people got this question correct on average. So that's the ones we'll be looking at for this. And I chose all of them that were under 50% for every chapter going forward with the reviews, so that, so some chapters, because they're more difficult, I might have more to go over. And some just have like one thing. Some people just get some stuff. So I don't know what it is, but we'll go over those things that were difficult. We do have some for today, since it's basically 3 chapters that we're looking at. And then of course, if you have any questions, which odds are you won't, but that's okay. We'll take those at the end. If you have really anything that you don't understand what I'm talking about during this, of course, you can always stop me. And I have the thing pulled up where I can see if you're chatting this time. It doesn't ding. I need to like set it so it has a notification so I can hear it, because I never see it when I'm talking. So first is chapters 1 and 3. The key concepts that you should have gone over, especially if you guys are hybrid. I don't know if I have any of the hybrid students in here. So I haven't really quite learned who's in what sections. I recognize names, but it's like all together in one big mush right now. But anyways, any of you hybrid guys, if you're here right now or watching this, Hybrid folks, you have a tendency to put off literally everything until the day it is due. If you haven't been paying attention and this is the first you're hearing about this course, which probably isn't because I'm going to talk to you when we get into lab on Monday, but heads up, that is a horrible idea in this class. Like, you couldn't have picked a worse class to wait till the last second to do everything. You think I'm joking? There's a reason why my hybrid students make 15% less average on their final grades than every other section that I have. Because people say that it's easier because they can just take their time and do it whatever and whenever and whatever. They're totally self-motivated and they can do this. And it turns out most people don't even know what that means to be self-motivated. And they think it means put things off until the last minute and I can handle it. And this class is not memorization of terms based. This is a class, and you'll see when we go through this review, if this is news to you hybrid people, that this is a concept-based class. It's an understanding and an application-based class, not a memorized words class. Those of you who are in my traditional lectures have really already picked up on that quite easily. So hopefully this is helpful to you online people if this is the first time you're seeing anything. You should be keeping up with the schedule that is given to you, same as the schedule that the traditional students follow. You should do your best to try to follow that. It will be good for you, I promise. That was loud. Okay, anyways, so chapter 1, really actually doing it now. Chapter 1, key concepts, the things that we talked about in chapter 1 alone. We talked about what microorganisms were. We talked about acellular microbes. Remember, acellular means they are not made of cells. So the acellular microbes that we learned about, just FYI, were viruses and prions. If we're not talking about a virus or a prion, it has cells. If we are talking about a virus or a prion, they never have cells. They are not made of cells. They are not cells, no cells for them, okay? So how do they reproduce? Basically by going into your cells and stealing and borrowing against your will all of your cells' machinery to do their thing. And we'll talk about in chapter 7 why that's bad. But some people were confused about how that works and what acellular means, because to a lot of people, if it is what we would consider alive, if it's like, you know, multiplying and stuff like that, it's cells, right? But it is not. You're wrong if you think that. Viruses cannot multiply without stealing your machinery, without living in something's cells. So because of that, we do not consider them alive because they can't live without that stuff. They're not dead. That's not the word for them. They're just not alive. And they are not made of cells. So cells are, you know, discrete items, if you will, membrane-bound items that contain everything that they need on their own in order to reproduce on their own without stealing any of that machinery they need to reproduce from anything else. They can do it on their own. They have everything they need. And when they make a copy of themselves, it has everything that they need. That's a cell. That's the definition of a cell. Okay, so that we're clear on that. That's why viruses and prions, again, chapter 7 will clarify what they are. I don't know if we have any questions based on those, but I just wanted to be sure because I had several questions about that we talked about that. So eukaryotes, anytime you have an A in front of something, in case you haven't picked up, it means without. So acellular, no cells, without cells. Eukaryotes, without karyo. No karyo. Karyo means nucleus, okay? So now we have cells that just have everything chilling in them with no compartmentalization, no segmenting things off. It's literally just a membrane enclosing stuff flowing around everywhere. There's no nucleus, there's no mitochondria, there's no endoplasmic reticulum. Anything that would have been, you know, made into an organelle and sectioned off, it does not have that. But it's still a cell. So we have the things that are not cells, things that are very basic cells are eukaryotes. They don't have a nucleus or anything else. And then we have the ones that are very complicated with the parts. Our cells, we are eukaryotes. That U means true. Okay, so that's kind of the distinctions that we went through. So we would think that eukaryotes, because our cells, that's our first life form, would be a single cell organism. They're cells, but they are super basic, that they would have been the first things to evolve on Earth. living wise anyways, to living organisms. And then the more complicated eukaryotic cells came later. Remember that our Earth's atmosphere is made mostly by microbes. We talked about what pathogens were, microbes that cause diseases. We talked about germ theory and Louis Pasteur. You do need to know Louis Pasteur's name. I would also be aware of Lister's name. It is on the list. So yeah, you would just be able to know what they did. So remember, Pasteur was involved in germ theory. He used those flasks to show that spontaneous generation, life just coming out of nothing, was a fake BS idea. And then also showed that there is something that they couldn't see at the time that had to be causing life to come out. Those microbes that they call germs. So that's why it's called germ theory. And that's why we still call them germs today. So Pasteur discovered that with those flasks that we talked about. Germ theory was that aseptic technique spawned from germ theory. Basically now, oh, wow, okay, there is this stuff that we can't see. So when we are going from, dead, rotting corpse and handling these birthing mothers, no wonder they're getting sick. There's something we can't see on our hands that we're transmitting to them. Okay, so now we know that that's germs. So how do we contain that? Aseptic technique. So if you ever think of sepsis, and that's whenever most of us are aware what sepsis is, right? If you are septic, or even think of a septic tank, that's where just tons of bacteria are breeding. So if you're septic, that means they're breeding inside your blood. You're going through sepsis. That's septicemia. So we know that that's got a lot of stuff growing in it. That's sepsis. So A is without, so that's without sepsis. So without things growing. So you're trying to prevent contamination. You're trying to prevent the spread of germs. And so aseptic technique, which is what we'll also be doing in lab, is where you're trying to prevent that spread. And Joseph Lister, where Listerine comes from, is the guy that first applied that into operating rooms, saying we need to wash our hands and we need to clean the actual surroundings to get rid of microbes so people aren't getting sick during surgery. All right, taxonomy, just know them in order, guys. I'm not going to ask you details about these. I would know domains because that is something that we broke down, and they are determined by first. This is where I wish that I had my thing linked. Let me pull up my pen, and I'm going to try to do this with my freaking mouse. I have a big mouse, it's a gaming mouse, so it's like really hard to maneuver, but we're going to try. Okay, so you have, actually, let me back up. Of course, I had to do that. Let me have to click on the thing. Okay. Okay, you have what we would call microbes. Okay, and these microbes, again, anything that we're going to be studying in this class that is too small to see with the naked eye, right? Microbes can be put into two groups, which we already talked about. This is going to be the ones with cells. I'm going to take forever. I always have to go back and explain things when I said I wasn't going to do it. Or things that are acellular. Which, remember, that means no cell. We'll just say this so that you guys can remember. So, and then we're going to go like this and do that. So you know that's what acellular means, in case you forget. Right. We already talked about the acellular ones, right? We said that's only going to be these two things. Viruses, and then prions or prions. I'm never going to discount you guys for mispronouncing things, okay? As long as you get the concepts. Okay, that's that for the acellular guys for now, right? Chapter 7, like I said. But for the cell guys, this is where our domains are going to come up. That's how we're dividing these cell guys, okay? So the first things we're going to do is divide them by whether they have a nucleus or not. And so that is going to be the eukaryotes. Please just understand that that means eukaryote. All right, and then this is the eukaryotes. So these guys, yes, have a nucleus. And these guys have no. And I'll write nucleus on this one so you know what they both are talking about. Oh my gosh. Anyways, so Right. So you either have a nucleus or you don't, but both of these guys are cells. They can divide and exist freely on their own, single cell or multi-cell. I don't care about that right now. Just whether they have a nucleus or not. And if you do not have a nucleus, if you're an eukaryote, you not only don't have a nucleus, you don't have any organelles, which are those compartmentalized things like mitochondria, chloroplasts, and a plastic reticulum. I know you've heard of them. Chapter 5 for that, okay? So anyways, so we've got these, you're either eukaryote or you are akaryotes. Turns out this is a domain. And then if you are breaking these, because this stuff, domains, are only done with living things. The acellular guys are not technically living. I'm not interested in putting those in domains and stuff, okay? So the cells are the ones you're going to be focusing on for domains. A D for domain. It means domain, okay? Let's be adults here. So eukaryotes, they do not have a nucleus. There's two groups that'll fall into this. I know I'm writing over everything that's over here, but just bear with me because I write big. We have the bacteria, which is going to be chapter 4, our next chapter. Gosh. And then we have the archaea. Okay, so this is a domain. I'm circling the domains for you guys because there's only three. There's only three you have to remember, but you do have to know them. I'm not going to make you remember the kingdoms, the animal, fungi, plant, all that. I need you to memorize those, but I do want you to know these three domains because it applies to our understanding of the differences between the cell types. We're not going to talk a lot about archaea in general. We really aren't, very rarely. So focusing on knowledge about bacteria and eukaryotes for this course, but still understand where the archaea fall in all of this. So first, if you're a microbe, are you made of cells or not? Second, if you're made of cells that can divide and make their own living cells without stealing parts from somebody else to do it, those cells can be divided as basic, general, no nucleus, no frills, nothing, empty sac of membrane and the junk in it. Or eukaryotes, nice compartments, including the nucleus, mitochondria, and all of that. Those ones without the simple cells can be divided as bacteria or archaea. Right, so you can think of the cells as being one of two things, with or without a nucleus. But if we're talking about domains, it divides it just one step further, bacteria or archaea. That's where the three domains come from. Bacteria, archaea, and eukaryotes. Three domains. Okay, so I hope that that made some sense. And then binomial nomenclature, we talked about this, like when we're talking about E. coli, you're just going to abbreviate it as big E dot little c coli, like we're used to seeing it. That's the most common one that everybody's familiar with. You are right when you say, oh, the E was big and the C was little. You are correct. That it is. That is the correct way to put that abbreviation. It's an abbreviation, though. The E stands for something. I won't make you memorize it, but it does stand for Escherichia. So that's the whole name, Escherichia coli. E. coli is just the abbreviation. So if you were writing out Escherichia coli, then your capital E is still a capital E in Escherichia. And your coli still has a lowercase c. And if you're typing it, italicize it. If you're writing it by hand, underline it. I'm never going to test you on the writing it by hand. There will be some circumstances, including unit exam 1, where I'm going to make sure that you understand the rules of binomial nomenclature with the italicized, with the what's uppercase, what's lowercase. Okay. Remember, the first word is actually the genus. When we're talking about kingdom, phylum, class, order, family, genus, species, the first word, Escherichia, is the genus. The second word is the specific epithet or the species identifier. Right, so that's what really is talking about the species as opposed to the group. If we're talking about something like salmonella, salmonella is a group of bacteria of Salmonella enteritidis, Salmonella typhi, Salmonella typhimurium, all this stuff. If we're talking about staph, it's Staph aureus, Staphylococcus, right? Staphylococcus aureus, it's Staphylococcus epidermidis, and et cetera, and et cetera, and et cetera. And there's so many of these. There's so many of all of them. But that Staphylococcus, that Salmonella, that Escherichia, that Streptococcus. All of those are the genus. So you have a whole lot of things that can fall into that genus. And sometimes we'll be talking about a genera, which is the multiple for genus. Genuses is genera. We're talking about several genera because some genera have shared traits. So do understand what the genus is in the terms of the binomial nomenclature, because it's going to help out with understanding in the future. Okay, that took longer than I meant for it to. I apologize. All right, chapter 3, what did we talk about? The 5 I's for identifying the organism. I'm really going to try to stick to just listing these. If you guys have specific questions, let me know. The differential versus selective media, selecting for or telling the difference. Enriched media, which has the growth factors that the fastidious organisms need provided to them because they can't make it for themselves. The enriched media has that. All right, microscopes, we talked about, we did kind of go over, it says we have to know the parts. We didn't really talk about it in lecture, but we're going to talk about it so much in lab, it's going to make you sick. So that's coming, okay? But when we get to those, and that'll be before we get to the Unit 1 exam, no worries. So real image versus virtual image, know which lens is giving you those. So remember the eyepiece, the ocular lens is the virtual image. You want to say it's the real because it's one your eye is seeing, but you are wrong. The one closest to the specimen is creating the real image. That's the objective lens. All right, anyways, oil immersion to prevent light scattering, heat fixing to keep your organisms killed. You don't want them necessarily alive all the time. And to make sure they're adhering to your slide, and as necessary, preserving what's in the cell so it's locked in place. Staining, there's negative and there's positive stains, okay? Acid stains, and I just had a question about this because it was on the quiz for chapter 3. If you haven't taken the quiz, you guys, I guess, are getting a little bit of an advantage here, but I had a question, and I usually have at least one person who's paying attention come up with the question of what this is what's going on here. So negative and positive stains. We talked about how the surfaces of cells have a negative charge because of the phosphate groups on the outside of the membranes. You got those hydrophobic tails that don't like water sticking in with each other, but on the outside are the heads for that lipid bilayer, and they have a negative charge. So the surface of your cells, generally speaking, have a negative charge to them. Negative doesn't want to hang out with negative. If you're a nasty person, you don't want to have nasty people hanging out with you, validating that you're both nasty. Okay, that's the idea. Negative, don't hang out with negative. For some reason, they're toxic, and the positive people are who are going to hang out with them. So it's a good example, I think. So when you're looking at this that way, negative doesn't want to hang out with negative. Which one do you think stains the cells? Do you think the negative is going to stick to the negative cells, or do you think the positives are going to stick to the negative cells? It's the latter, right? It's going to be positives, positive charge, opposites attract, stick to the negative cells. The negative stains repelled by the negative cells, so they're going to stain the background. Okay, so that's why they work the way that they work. But our lecture also talks about acid and base stains, right? Acid and alkaline stains, or acid and basic stains. And we learned in chapter 2 that an acid is something that has a bunch of H pluses. And if you're giving up a bunch of H pluses that are positively charged, wouldn't that be a positive stain? But it isn't. Because yes, the H, so if we're talking about, I'm gonna write with my pen and I forget I can't do it. If we are talking about, for example, H, and then it's attached to, I don't know, whatever your stain is. We'll just put an S for now. It's whatever your chemical makeup is of your stain. And that H in its solution will give off the H pluses, just like acids do. Throwing off that balance, you know, of the H pluses and the OH negatives in water should be equal. Not anymore with this. Okay. Yes, the H pluses are positive, but when the H pluses are positive, what are the S's?
Negative.
Right, negative, exactly. So we've got a negative charge to the actual stain molecule. So that's what this is talking about. When you hear negative stain, the stain itself is negative because it gave up the H's, which were positive. So that was an acidic stain. So that's why they are what they are chemically, so that you understand the concept of why those things go with what. We understand negative stains and positive stains and how they work with the cells. So if you were ever asked about which one is acidic and which one is positive, just try to think back on, we're talking now about the molecules of the stain. Okay, not the H pluses or whatever accessory molecule it came with. Right. All right. Differential stains. These are just to help us tell the difference between the things. We're going to go over those in detail in chapter 4 and in the lab, and I'm not going to cover them here. Again, I know, once again, I'm not going into detail over them. All right, chapter 2, the next chapter that we really went over on Wednesday or Thursday. Chapter 2, we talked about the types of the bonds. Remember, I said covalent. If this isn't the good example, then I don't know what is. If my sister and I have to share Barbie, that's covalent. We're sharing the Barbie equally. That's the electron. Sharing the electron, sharing the Barbie, same thing. Am I going to hog that Barbie? Heck yeah, I'm going to hog the Barbie because I'm a jerk, okay? Because I am oxygen, and oxygen is a nasty jerk, okay? So oxygen's the nasty jerk. It's going to hog the Barbie, but because we have a covalent bond, which is essentially happening because mom is watching us, we have that covalent bond. I still have to share with my sister sometimes, but because I'm hogging that electron, that Barbie electron, that makes me have a negative polarity and makes my sister, who's crying because she's not getting the Barbie as much, have a positive polarity, even though we're still sharing a little bit. Yeah. So that's when we have electronegative atoms hogging things, creating polarity with covalent bonds still. Sharing, but hogging. If you're a carbon and you're happy and you're chill with everybody, and you're sharing equally with everybody, then there is no polarity. Everybody's equal. There's no charge at all. And charge, think of it as drama likes drama. Drama breeds drama. Think of the charge as drama. So all this charge going on with the water, having its, moments of like positive and negative polarity, all the things that are associating with the water, with their positives and their negatives and their polarities and their charges and everything. That's a lot of drama going on. Everybody associating with everybody, everybody doing their own thing. Whereas carbon and hydrogen are over here chill. They don't need to get in that mess. And they don't want to. And the mess doesn't want them to come in there because they're happy being a mess. So that's hydrophobic. Carbon and hydrogen, they don't want to deal with that. Hydro, the water, charges, and everything that comes with that. No. And the hydrophilic is everything that wants to get in with that water and its charges and its drama. All right, so me, I'm dramatic apparently because I'm the oxygen here in this. I am dramatic, let's be honest. So I'm going to be electronegative. I'm going to be hogging the Barbie. And sometimes, as water will like to do, sometimes I'm going to run off with that Barbie and it's mine now. So I'm leaving my sister alone crying in the front yard. I mean, I grew up in the 80s, so everything is outside. So we were outside playing for sure. So I'm going to leave her in the front yard and I'm going to go run down to the neighbor's house because again, in the 80s, everybody played with everybody's neighbor's kids. And we'll just show up over there. And so we would, so whatever, I might run off with that Barbie and go play with it on my own, or I might, play with somebody else. But the idea is I've run off with that Barbie. I stole that electron and I left my sister without any electron now. Hydrogens only have one. She only had one Barbie. She was, Barbie poor. So she only had one Barbie and she had it stolen from her. So now she's left with a positive charge because that electron was negative. The negative was stolen from her. And now she's a positive. We call H pluses hydrogen ions or protons because of that. Anyway, so my sister's a proton now, and I'm running around with this electron doing my own thing. And now that we are not sharing the Barbie anymore, the electron, we're not sharing it anymore. I stole it because she donated it to me, right? That's an ionic bond now. Hydrogen can do that on water. With that OH group, they can separate off and become the OH negative and the H plus. Like we said, Schrodinger's cat kind of happens just all the time in water. It's just constantly going on. Whether we're going to be covalently bonded or separating as ions, that's a unique trait from water that makes it the solvent of life. Its ability to do that. Its polarity and its ability to sort of become H pluses and OH negatives, right? Hydroxyl groups. So that's the concept of water. Why is this solvent? That's the concept of where polarity and hydrophilic come from. That's the concept actually too of where pH comes from, because if you have H2O, so it's two H's on an O, And one of the H's is kicked off and the OH is running off on its own, right? That concentration of the H pluses versus the concentration of the OHs, if you have pure water, should be equal. And that's pH 7. And so if you have too much of the H's, now we're throwing off that balance. That's an acid. If we have too much H's, way you can remember that is if you know that hydrochloric acid is an acid, that H is there, right? We're adding in H's into a solution. That's what's making it acid. Is there OHs there? No. So is there in sodium hydroxide now? Yeah. So the hydroxide atoms can be added in to the water where we had the H pluses and we had the OHs that were equal. When you add in more OHs, those H pluses that were equal are now going to get more attracted into the the OHs, and we have less H's floating around. So that's how come that decreases because of that. Anyways, so that's how that balance works out. Remember, 0 is going to be acidic and 14 is going to be basic because 7 is neutral because of the equal 10 or so whatever. But the numbers, what they mean is the reason that it's a lower number for more H pluses is because we're talking about the negative logarithm of the concentration of the H pluses, which literally just means 10 to the negative whatever your pH number is. Because obviously 10 to the negative 1 is a bigger number than 10 to the negative 14, right? So think about it that way if you need to in order to determine, does it have more H pluses or less H pluses? Because that 10 to the negative number is what's determining if it's more or less as far as the concentration of the H pluses. And then that number that you had put in the top, the exponent, that -2, -1, whatever it is, that is pH of 2, that is pH of 1, or pH of 14, it was studying the -14. And that's why it is basic, and that's why it is acid, and that's why the numbers are the numbers, right? Cool. Did it again, said I wasn't going to do it. The biological macromolecules, there's 4 of them that you need to understand kind of what they are and what they're made of. The carbohydrates, the lipids, mostly hydrophobic because those carbon hydrogens, right? Remember our amphipathic, which amphi means both. Their amphipathic molecule that's really important for biology is phospholipids. The phosphate group with all those, the P with all the oxygens around it. Didn't I just tell you how nasty an oxygen was? Well, they got so many freaking oxygens around. We got 4 freaking oxygens around it. That 1 P, the phosphorus isn't as important. It just allows there to be 4 oxygens around. So all those oxygens are what's the nasty. That's why they're so negative on that phosphate group, anywhere you see it, whether it's ATP with the triphosphates or it's a phospholipid. Those phosphate groups are so negatively charged. And then when you stick on that nice chill carbon hydrogen, you've got both going on. You've got the hydrophilic in the charge, and you've got the hydrophobic in the tail. And that's what builds our membranes, right? Anyways, so that's our lipids. That's the concept of they can be hydrophobic all over, or they can have the amphipathic quality. Proteins, amino acids are making up the proteins. Know the 4 structure levels of proteins. And then the nucleic acids, RNA versus DNA, and then ATP. And those are made up of nucleotides. That's their subunits. So yeah, cool, good. Okay, now we get into the missed concepts. We're going to have a slide per missed concept, so hang in there with me, okay? Chapter 1, germ theory. I've spent more time explaining it in this semester than I usually do because of this issue. But in case you missed it, germ theory is developed by Louis Pasteur with those flasks that we saw with the little necks. And based on if you cut the neck off and it got more exposure to microbes in the air versus had the long neck where the microbes couldn't get in as easily and wouldn't grow, he showed that life doesn't come from nothing. It doesn't spontaneously generate. It has to be induced by something, even if you don't see it. And that was germ theory. The germs are what are doing that. Louis Pasteur, okay, he did that. And yeah, you'll get asked about him and you'll get asked about Lister on the exam. Okay, so I have mnemonics. They're not always actual mnemonics. A mnemonic is like whenever I was telling you guys about kingdom, phylum, class, order, whatever, family, genus, species. Writing, I would have to literally write it out when I said the words because that's how I remember it. But some people remember it as like King Philip could whatever, whatever, whatever. And then you could say, okay, King, K, Philip, P, C, could, and then write the letters down that way. That's a mnemonic. All right, so some of these are actual mnemonics and some of these are just memory devices, but they're always going to say mnemonic on there because I'm lazy. So my memory thing here, pasteurization gets rid of germs. You know, if you have pasteurized milk, we've gotten rid of at least some of the germs that are in your milk, so it'll last longer than it would if it was not pasteurized. How much longer does ultra pasteurized milk last than regular milk? Have you guys ever had Lactaid or anything like that, lactose-free milk? That milk lasts forever, and I'm not kidding. It's like 2 months or more, even after you open it, because it's like ultra pasteurized. So they've killed even more of the germs. And to me, it tastes the same. And I'm not even lactose intolerant, but I buy it because it lasts longer because I don't drink a lot of milk. So FYI. But pasteurization, yes, he came up with it to get rid of the germs that he realized were there because he's smart like that. Okay. So pasteurization gets rid of the germs, pasture, germ theory. All right. So here's some questions. These are not, by the way, these questions I'm going to go over, not the direct questions from the exam. They're just meant to help you put this into a context. So first question, who developed and tested germ theory? So if you were to look through these choices, of course, when you guys come back, if you guys want to come back to the slides later and just like look through the slides and see if you can still understand why the correct answer is the correct answer. Remember, I told you that's more important than knowing the correct answer, is understanding why it's the correct answer and why the other ones are wrong. For example, this is an easy one to do that with. Pasteur, we know that's the right answer because we know he did germ theory. But even if you did know that, just because you memorized it or whatever, let me tell you why the others are wrong. It's not Lister, because Lister did the aseptic technique in the operating rooms and washing hands and stuff, right? Listerine guy. It's not Einstein, because that's physics. And it's not Jenner, and this is not talking about Kylie Jenner or something. There was a guy, Edward Jenner, who developed the very first vaccine for smallpox. Okay, and we'll talk about him a little bit later because he had a whole other thing going on. First vaccine ever for smallpox, one of the worst diseases that's ever existed. It has been obliterated from the planet completely now, thanks to his vaccine. Why is a vaccine called a vaccine? Vacca is the Latin word for cow, because Edward Jenner realized that milkmaids weren't getting smallpox. They had beautiful skin. They weren't getting smallpox. So what he did was scrape off the lesions of cows that had cowpox and then scraped that into his little boy's arm and then pushed him out into the world of smallpox. And he didn't get smallpox. And that is legit how the vaccine works still today. It's just now, not forcing little kids against their will and medical, experimentation. Obviously a long time ago, but that's why vaccines are called vaccines, because it came from cowpox, because what he did, Jenner. Freaking nuts. So the next question, I try to put like one simple question and kind of one more complicated question. Plague doctors, we went over this in class if you were in lecture. Plague doctors stuffed the nose of their masks with herbs and flowers to ward off bad smells. They thought the bad smells were causing the spread of plague. Why is that ineffective? So we'll go through our choices. This would have worked if herbs were antibacterial. Smells do not transmit illness. It blocked them from seeing their patients clearly to treat them effectively. That's why it was ineffective. Or the contaminated herbs were being transported from home to home. Now, there's some of these that you might want to lean into because they make sense with a little bit of logic, but then they break down if you really think about them. Obviously, I want to say obviously, maybe you don't know. B is the correct answer. Smells don't transmit diseases. Miasma was that theory. Smells don't transmit diseases, or you would literally be getting poop diarrhea every time you walked into somebody's bathroom or in there. So that isn't how it works, clearly. And are they associated sometimes with bacteria being there? Of course they are, but it's usually volatile compounds that bacteria are making, right? They're just making bad chemicals that smell bad to you. It's not actual disease there. So they were stupid about that back then, clearly. But so A, this would have worked if the herbs were antibacterial. The bacteria that causes the plague weren't getting in from the smells, and they weren't getting in from the noses of the masks. And if you know anything about the plague, you probably would tell me how it's spread, which is through the bite of fleas. So that doesn't have anything to do with any of that. So it's not A. It blocked them from seeing patients clearly. That doesn't have anything to do with what we're talking about. It's not C. And then D, the contaminated herbs were being transported from home to home. Well, I mean, I can imagine if smells had illness in them, that, yeah, the herbs would get contaminated. And then you would be taking that illness in the herbs from home to home. But that isn't how it works, right? So that's not why it was ineffective. It was ineffective because smells don't transmit illness. Cool. So that's the kind of thing I'm talking about. Like, think through your choices. All right, and the next concept. These will probably go a little, I say they're going to go faster. You guys have met me. All right, cell evolution. Eukaryotes lack a nucleus without nucleus, right? And then the other ones, the eukaryotes with a nucleus and all the other nice parts of a cell are more complicated. So the less complicated generally comes before the more complicated. We talked about something called endosymbiotic theory of where those organelles came from, which we thought was other bacteria. We will go over that in more detail in chapter 5. So if you heard me talk about it in class, don't lean on it too much right now because we'll get into detail on it in chapter 5. All right. So eukaryotes don't have any structures going on with them. The eukaryotes do. So the eukaryotes are more primitive, they're more basic, and they likely came first. So here's a way to remember that. A comes before E in the alphabet, right? So eukaryotes become before eukaryotes. So the eukaryotes probably came first in evolution. Sample questions. It is believed mitochondria, our powerhouse in our cells, the eukaryotic cells, evolved from a bacterial symbiont, like I just said. If you can't have eukaryotes without mitochondria, and folks, you can't. If you sat through any of my lectures this week where I told you how I was going to suffocate you and you would die because you aren't going to keep up enough energy in your body, in that short amount of time, you'll use it all up if you don't get oxygen to help you deal with that, basically. All of that happens in your mitochondria, our powerhouse. That's where your ATP is made. That's why we call it the powerhouse. So all cells that are eukaryotic cells have to have the powerhouse because they have to have energy. So if you're eukaryotic and you're compartmentalizing everything, you've compartmentalized that energy into the mitochondria. Again, we'll talk about it in more detail in chapter 5 because that's eukaryotes. But if eukaryotes can't be without the mitochondria, and we think that the mitochondria evolved from some sort of bacteria back in the day, what can we say? Either A, bacteria evolved from eukaryotes. So in that line of thinking, that can't be correct. B, eukaryotes existed before bacteria. Eukaryotes didn't exist until they merged with the bacteria, so they couldn't have existed before. Some other thing did, maybe, but not eukaryotes. Bacteria existed before eukaryotes. Now we're talking, right? And then D, eukaryotes evolved from archaea. We're not talking about that. So if you remember your little setup, you remember archaea are separate from bacteria and from eukaryotes. They're not in the equation here, right? So that's how we get to that answer C, in case you weren't paying attention. Oh, looking at a microscope, you see a large cell with several membrane-bound sections. What type of cell could this potentially be? From our choices are yeast, archaea, bacteria, virus. If you've been paying attention, this should be an obvious one. What did we say about archaea and bacteria? What category did they fall into? Eukaryotes, no nucleus. If they don't have a nucleus, they don't have any membrane-bound sections. So it can't be B or C. D, acellular, you have to have a cell in order to have cell parts. Can't be D. So even if you didn't know yeast were eukaryotes, you learned it just now because it can't be the other thing. Process of elimination, right? Which we will bring up more specifically in chapter 5 again. So chapter 3, we talked about selective and differential media. And I tried to go in a little more depth in it, but remember selective media, we are selecting for certain things to grow. Differential media, we're trying to tell the difference between things, but it doesn't affect what can or can't grow. You can have both of those at the same time though. So media that selects for staph and can tell the difference between Staph aureus and Staph epidermidis. Now, wouldn't you rather know, if you're working in a hospital lab, the difference between tame staph epidermidis and disease-causing staph aureus? Yeah, and they have media that does that for you. So it would be quite useful in diagnosing some things when you don't want to wait on culturing something and then waiting on the PCR. PCR is not as fast as you think it is every time, okay? And all of that. Plus, if you're trying to determine if something is antibiotic resistant and all of that, you want to be sure you're isolating the right thing that you're testing, right? And then once you've isolated the right thing that you're trying to test, of course, you might want to run it through a couple more tests to double check and all of that. So all of these tests are sometimes just like double checks, as opposed to just this one thing, this one media that we use decides definitively if it's positive that this is the thing. When I worked in food testing, this is why I want to prove you wrong with this. When I worked in food testing, if we had PCR positive for E. coli or Listeria or whatever, let's go with Listeria because that was the more common one. And Listeria is what's bad for you if you're a pregnant woman eating deli meat. Okay, that's the one they tell you to heat up your deli meat. So Listeria can cause meningitis and it can cause horrible GI symptoms and it can be abortive. That's the worst of it, right? So abortive or stillbirths and things like this. So bad. Don't mess with it. It's sort of like the kitty litter thing. So people think that that's like not that serious, but it's like 30% fatality rate for a fetus. Don't mess with it. Okay, had somebody else do the kitty litter. And most cats have it. Most people have had it in their lifetime and they just didn't know. But when you're pregnant is when the problem is. So we'll get to those later. I know you're excited to learn about it. Geez. So anyways, we're talking about these different kinds of media. And when I was in that lab, and if I was testing for Listeria and the PCR said it was positive, we didn't just run with that. What if we had contaminated it with something in our lab that had Listeria in it? Because we were running thousands of samples a day. So we would take sample from that original food, and then we would run it through all of these different plates that we're going to be learning about, the different types of plates, the different types of biochemical tests, and all of these things to just verify after step after step after step that this was in fact Listeria. And I mean, there was lots of steps. So whenever you are diagnosing somebody, it's the same thing. You're not going to run necessarily one test, especially whenever somebody's life is actually on the line. You want to be sure that you're doing it right. So that's the idea with selective and differential media. Selective, we're going to select just for the target audience. And then differential media, telling the difference between things, whether or not you selected first. Doesn't matter, you can still use it to tell the difference. That's usually color changes. And color changes occur from differences from breaking things down. So if I break something down differently, I might make this product, this acid, but you can't break it down at all. So you're not making the acid. So we might have a pH color changing indicator in the media to tell you if acid was made or not. Because if it turns yellow, that means acid was present, and that means this metabolic pathway. Doesn't stop it, just, oh, it was yellow, check. I understand what it means. It's usually biochemical pathways. All right. So, which of the following is an appropriate definition of selective media? Media that changes colors due to metabolic byproducts. Media that favors the growth of some organisms and inhibits the growth of others. We saw that specific choice when we did the Kahoot, if you've done the Kahoot. Media that selects for organisms which require added nutrients. And then media that kills all bacteria except for one type. So these are very similar to what we saw in the Kahoot questions. Even though I tried to write them from scratch, just I guess works out that way, so mine works. So let's look at C first. All right, media that selects for organisms which require added nutrients. It has select in it, and I told you selective media selects for things, but we're not talking about added nutrients, whether you need an extra nutrient or not here. We're talking about selecting for an entire population of bacteria and selecting against several populations of other bacteria, right? So we're favoring this group here. That's what selective media does. So that's why B is the answer here. I think you guys know kind of why the other ones aren't the answer, right? It doesn't kill all bacteria except for one kind, because you can have whole groups that can fall in. I have media that is selective, that only gram-negative bacteria will grow on, and gram-positives can't grow on. That's like a whole half of all bacteria. So clearly not just one type of bacteria going on there, right? Not one species or something. All right, next question. McConkey agar, we'll learn about that in the second half of lab, contains something called bile salts. Now, where do we find bile? In the GI tract, right? It's really that simple. So this will favor gram-negative bacteria. It will inhibit gram-positive. This is one of the medias that I was talking about. Again, see, I say medias and I don't say medium and media. Okay, so simultaneously, the media contains an additive that causes bacterial growth to turn pink if it can break down lactose. What type of media would this be? So let's look at this. Is it selective? Do we have a selective thing going on here? Yeah, because we're only letting the gram negatives grow. The bile salts killing off our gram positives and letting the gram negatives grow. That's selection. Okay, next choice. Is it differential? Well, we talked about this metabolic or biochemical difference. If you can use lactose or not. If you can, your thing turns pink from chemical reaction, or your thing can't use it and it does not turn pink. That tells you information right there, right? Whether you can use lactose or not. That's differential. That part doesn't affect who can grow and who can't. It's just telling you if you can use lactose. So we have both of those things going on there. So our answer is C. So if you guys come back to the reason I put Y down there, if you guys can come back and look at that and go through that question again and figure out why it falls into both categories, then you're really understanding the difference between the selective and the differential. Okay. All right, next one, growth factors. The growth factor is the thing that you have to have added in because the organism can't make it for itself. We cannot make vitamin C. We cannot make B12. We cannot make a whole lot of other things. Lysine, for example. Remember the lysine contingency in Jurassic Park? It's actually real for all large organisms. So they can't make their own lysine. We can't make our own lysine. So we have to have it in our diet somewhere. We don't really think about it. You might take a multivitamin and that helps with some of this stuff. But in general, you can get it from a healthy diet. Whenever you have, let's say you're not eating a healthy diet, let's say you're on a diet and you're very restrictive. Let's say you're doing keto or something like that, and you're not eating enough of something to give you this vitamin 2, we'll just say that's not a real thing. You're not getting any vitamin 2 because you're eating mostly protein and not enough carbs. So you're missing on vitamin 2, so you would want to take an actual vitamin that had vitamin 2 in it. Or you'd want to start supplementing your food with the vitamin 2. So imagine that these are bacteria that have to be keto. That's just what they have to be. They're giving them a keto diet to live off of. That keto diet that you're putting your bacteria onto on your media doesn't have any of the vitamin 2. They're not going to grow. They're going to die. So you have to provide vitamin 2 into the media. That's exactly what this is talking about. Vitamin 2 is the growth factor. The media supplying the vitamin 2 is now called enriched because it's been enriched with the vitamin. It's been enriched with the growth factor. Vitamins for us, similar thing to growth factor, but growth factor doesn't mean vitamin. Because even I just said lysine earlier. That's an amino acid. That ain't a vitamin. So that's also a growth factor. That's just an example, okay? So don't forget that, but it's useful to think about it that way. So it's enriched because we've enriched it with the growth factor, whatever it is, and the bacteria that couldn't grow on keto without the vitamin 2, that bacteria is fastidious because it needs the vitamin 2. So hey, surprise, we are fastidious. And our media somewhere in our lives has to supply us our growth factor of, vitamin C or whatever. Okay, so that's the idea. So if we're talking about growth factors, and even if you have to remember about, you have some choice in your question, it's talking about growth factor versus enriched versus fastidious, okay? Growth is a factor you can't live without. So think about it that way. Growth itself, well, you have to be able to grow to be an adult, right? And if you can't grow, then you would stay a little tiny fetus forever. So you have to be able to grow in order to live. So growth is something you can't live without. Growth factors, therefore, are things you can't live without, right? If you can remember that, those are the things you can't live without. As far as being fastidious or enriched, I feel like enriched speaks for itself because you're enriching it with these good growth factors. It's being enriched. It's becoming rich now with the extra things. Whereas fastidious is just the word that's left over. It sounds kind of like it's, spoiled or something anyways. All right, so which scenario details features of a fastidious organism? So first you would want to know, what does fastidious mean in your mind? It means we need a growth factor, something that we can't survive unless it is provided to us. We can't make it, right? Can't make it for ourselves, has to be given to us. So bacteria that turn pink when they're growing on McConkey agar, like I just talked about previously, or cows, they're herbivores. Okay, is that fastidious? Velociraptors, which require lysine in their diets, which we've been paying attention, obviously that's the right answer. And archaea, which grow in extreme environments. So fastidious means you require the growth factor to be supplied to you because you cannot make it. Is that what A is talking about? No. A is talking about media where you turn pink if you use lactose. It doesn't mean you have to be supplied lactose because other things can grow on it and don't use lactose. It's not what's going on there. Cows, they're herbivores. What in any of that says that they have to have something specific supplied to them? It just means that they eat grass, but that doesn't mean that they have something like a vitamin that they need to supply to them. I'm sure they do, but that alone, that fact that we're talking about in B, don't read past it. The information I'm giving you in B, the fact that cows are herbivores, doesn't suggest fastidious, right? Okay, velociraptors, I'm literally telling you, they require something added into their diets. So even if velociraptors aren't real and we don't know that about real dinosaurs, I'm telling you that in C, this is the only one that is explaining a fastidious organism. And then archaea, that's nice for them. They grow in extreme environments. Cool. So, second question, which of the following could be considered enriched media? All right, enriched media where we're adding something that another organism needs to have it supplied to them so that they can survive. McConkey agar, biosalts will inhibit some things and favor other things. That's not talking about something that's needed. That's just talking about favors. So Simmons citrate. We have citrate as our only carbon source available, and only some organisms can grow and use that as their only carbon source. This is looking at it almost from an opposite view, right? This isn't talking about something that the organism has to have. It's talking about, we're only supplying you with this. Can you survive? It's that. That's not the same question. Nutrient agar, a basic medium. Most people can grow on it. Most organisms can grow on it. Okay, that's all that means. Is there anything extra added to it that, some things might need? No, it doesn't say that, does it? But blood agar has nutrients involved in the blood that some organisms need in order to grow. They can't make it for themselves. And that's what D is telling us. So D is the right answer here. Chapter 3, we have the differential stains. This is the gram positive, the gram stains, the acid-fast stains, the capsule stains. Anything that tells you anything about bacteria. Does it have this? Yes or no? Is it positive for that or negative for this? Anything that's telling you the difference when you're doing your stains is a differential stain. Just telling you the difference, right? All right, so here are some examples: the capsule stain, the acid-fast stain, the gram stain. And these are the questions they'd be asking. Does it have a capsule? Is it acid-fast? Is it gram positive or gram negative? Okay, so remember, chapter 4, we're going to get into the details of how the gram stain works. And we'll do that in lab as well. All right, some stains that are used in differential staining work on based on the charge itself. So that's where we were talking about, phospholipids having the negative phosphate group. That's on the outside of the cell based on the makeup of our lipid bilayer and how our negative and our positive stains work. I'm not going to go over it again right here because we talked about it in pretty good detail earlier on, all right? Another way that you can remember negative and positive stains and negative cells have negative attitudes, so they're not going to get along with one another. All right. So first question, which of the following would be considered a differential stain? So just telling you the difference between whether your organism has a thing or not. A, staining for the presence of flagella. That's our structure that helps the bacteria be motile, be able to move. B, a stain using one dye that stains the cells one color to create contrast. That's a simple stain. A simple stain inherently in its definition is not a differential stain, to be clear, right? C, a stain which kills the bacteria first. What does that have to do with telling the difference between things? D, staining a slide with one dye and another slide of the same organism with a different dye and then comparing them. No, you're just looking at that same organism in different colors. That doesn't tell you the difference between anything. But these staining procedures that we're going through depend on actual chemical makeup of your organisms or structures that your organisms have or lack. These stains work because of those aspects. They're telling you the difference because they're literally interacting with the differences. So A is our answer, whether you have flagella or not, okay? That's telling you the difference. Oh, sorry. The second question is mycolic acid. That's a wax on the surface of our acid-fast bacteria. That wax, all right? Why might you guess that a special differential stain, carbofuscin, which we'll use in lab, would need to be used to stain positive cells? They are acid-fast positive. Why do you need carbofuscin to stain them? Tell them from the acid-fast negative cells. Now, this doesn't tell you or imply that I even told you about how the stain works. I didn't, I think in at least one of the sections, but I'm going to go through it anyways. All right, how you could deduce this, even if I didn't already tell you the details about the acid-fast stain. A, carbofuscin must be a positive stain. Well, we know positive stains are going to adhere to the outside of cells. So it would stain cells, but it would stain any cell, right? It wouldn't have any favoritism for mycolic acid. So if it's just a positive stain, that's not helping anybody. B, carbofuscin must be lipid soluble. Lipids are anything that are, we're talking about the hydrophobic molecules here, the fats, the waxes, the sterols, things like that. Now wax, like paraffin wax, is hydrophobic, right? It's a lipid. Mycolic acid is a wax, therefore it is a lipid. And so if we're talking about something that's lipid soluble, we're talking about something that can actually interact with the lipid itself. So they can actually, commingle with one another. And so the carbofuscin mixes with the carbolic acid and they get into it and they just stay best buds. Even if you rinse them with acid to get it out, they're going to stay, because what does lipid not care about? Charged molecules like acid. Don't give a crap about an acid. Don't give a crap about an H+. Don't give a crap about a negative charge. But their lipid, the lipid, yeah, we're staying away from that other drama. And that's exactly what's going on with carbofuscin. Okay, so we know B is the answer, but let's see why C and D are not. Carbofuscin must be heat activated. What does that have to do? What? I mean, I guess that, you might, maybe it might. I don't know. We haven't talked about that, right? But what does that have to do with it, causing a positive or a negative stain on something? So that's not the thing that's telling us the difference with the presence of mycolic acid or not, which is what acid-fast stains are looking for. And then D, carbofuscin must be a negative stain. Same reason for A, that that's not the answer. So that's why B is the answer here. So even though we didn't talk about the exact steps of the acid-fast stain, you understand about chemistry and you understand why B is the answer. Again, another good one to come back to after you've done some studying. Right. Next one, gram stain in general. Yes, chapter 3 did introduce the gram stain. And I apologize for that. Only because it expects you to know the steps of the gram stain. It introduces it. We're going to go over it here. It might be even a good preface to learning the gram stain later on in chapter 4, our next chapter, or in the lab. And it does talk about it in chapter 3. So it doesn't really matter which chapter you're pointing your finger at, you're going to have to learn it, yeah? So here we go, gram stains. Crystal violet is the stain, it's our first stain. There's the steps. Well, actually, let's go through this. Crystal violet, iodine, alcohol, safranin, which is red, by the way. I know crystal violet gives away its color in the name. So crystal violet's a special stain. It is going to get stuck in the cell walls of the gram-positive bacteria. Gram-positive bacteria have a very thick scaffolding cell wall, very thick. And so the crystal violet, sort of like when we play like Plinko or something, I probably have no idea what I'm talking about. But It's sort of like if you had stacked mesh nets and you're dropping a ball down. Is it gonna just go straight down through? No, it's gonna fall through holes occasionally if it gets stuck up on certain layers. That's sort of like the peptidoglycan cell wall of these bacteria. Learn about peptidoglycan and that scaffolding of the cell wall in chapter 4 in more detail, but for now, just understand it's like a big, thick scaffolding around the gram-positive cells. And then we have gram-negative cells. You've already heard me say several times that gram-positive versus gram-negative is probably the biggest #1 difference you need to know about bacteria, because that is the #1 identifier between them. So gram-positives have this thick scaffolding. Guess what? Gram-negatives don't have the thick scaffolding. They have like a layer, like one layer of scaffold, like tables. That's it. That's like it. And because they don't have that thick scaffolding, that obviously it's thick scaffolding is going to give you some rigidity. You're going to have shape because that's surrounding your entire cell and protection and stuff, right? If the gram-negatives don't really have that, what do they have to help them have their shape and their, protection and everything? They have another membrane. They have 2 layers of lipid bilayer. So they have lipid bilayer, like tables. It's a thin peptidoglycan layer, thin layer, and then another membrane. So that's how they do their thing. Massive difference in the outside of those cells. So the gram stain relies on that difference. The crystal violet gets stuck in that scaffolding of the gram positives. It's trapped in there. The iodine locks it into place. I had said to you guys, mordant is the word for that. Think when you hear mordant, mortar. That's literally how I learned it. They're not the same word, but they have similar sort of uses. They're going to help things stick in place. Brick mortar helps bricks stay in place. The mordant of iodine is going to help the crystal violet stay in place if it got caught in that scaffolding, in that webbing, in the Plinko machine. Okay, so it helps it get stuck in place. It's not going to get stuck in place in that single table layer. It don't care about that, right? So you'll put your crystal violet onto your gram-negative cells. At face value, they'll be purple. But when you add your iodine to it, it's not helping anything stick in place. So after your iodine, it might look purple still, but when you rinse it with alcohol, removes, it's a decolorizer. It removes any of the crystal violet that didn't get stuck in place by the mordant. So if the iodine didn't work for you on your little single table, alcohol comes in, it dissolves your outer membrane, and it rinses out any of the crystal violet that didn't get locked in place. So now, suddenly, our gram positives are still purple because it got locked in place. But our gram negatives are back to clear, uncolored, because we've rinsed that crystal violet right out. That's what the difference is there. And then we counter stain with safranin because now you have colorless gram negative, and it's really hard to see colorless. So we still want to be able to see them. So we do safranin to turn it red. To me, gram positive, purple. P positive, purple. To me, red is a negative color in general. So I think red, negative. The books and descriptions are always going to call safranin pink. It looks more pink when you look at it in a microscope, okay? But if I'm talking about trying to tell the difference between these things, and we know purple starts with a P, and I know, pink might too, but red doesn't. Purple ain't going to be mistaken for any other color. And believe me, when you look in those microscopes, the purple is purple. There is no mistaking it once you see it. So remember, purple positive. Ain't purple. It's negative for the gram stain, okay? If it's red, I think negative, okay? Example questions. What is the difference in a gram positive and gram negative bacteria that makes them stain the way that they do? Is it differences in the charges on the surface of the cells? Is it the presence or absence? Yeah, I go inoculate the cultures, I know. Is it the presence or absence of LPS? I didn't even talk about here. So let's just leave that for now. Presence or absence of a nucleus, or differences in the bacterial cell envelope. When we're talking about the envelope of anything, we're really just talking about the outermost layer. So Gram-positive envelope, thick peptidoglycan. Gram-negative envelope, very thin peptidoglycan and an extra membrane. And that's what's determining the difference here, right? So you don't even need to know what LPS is at this point. All right, the next question, a patient sample is sent to you testing for the presence of a suspected gram-negative bacterium. After performing the stain, the cells are purple in color. What are you going to tell the medical team? A, the patient sample is in fact gram-negative. They are correct. Purple is positive, so it ain't A. The patient sample is gram-positive. They were incorrect. Okay, B, let's see what C and D say. C, they must have sent the wrong sample. D, the gram stain is inconclusive. Well, they thought it was negative, right? And it's staining purple. So maybe you were wrong. So it's probably inconclusive. So you're going to need to repeat it with a fresh stain. No, if it's, you did your gram stain and you know how to do a gram stain and you're following all the rules here, you don't need to redo it. You're not wrong. You don't need to do any of that. B is your answer. Okay. This isn't about asking a question and being unsure. Shouldn't be any asking questions and being unsure in medicine. Why is this not going? Okay. All right, chapter 2. Most missed concepts, starting with chemical bonds. Man, if I haven't gone over these Barbies enough with y'all. Chemical bonds, covalent, we are sharing. We have somebody hogging the electron. The oxygen is nasty and it's going to hog the electron, just like I was nasty and I hogged the Barbie from my sister, older sister, so I do things like this. Ionic. This is when one atom donates its electron to the other. Yeah, it makes the outsides of the atoms happy, but it's a better story to say that I stole the Barbie from my sister completely and just went off and didn't play with her anymore. So this is when you're literally just taking the electron away. You're not sharing anything anymore. You just took it. That's ionic bonds. And because you have an actual charge to your ions now, you are separated completely. Just like when OH just decides it's stealing H's positives when it's in water, that OH, that hydroxyl group, has this negative charge because it took that electron with it. That H, the H plus ions now, those protons that are floating around now because they had their electrons stolen, their Barbie stolen from them. They're just floating around like that now. They ain't sharing anything with anybody. So those guys are separate. They're not exchanging, but you have a whole bunch of OH negatives and H positive mixed in together near one another. They're still going to be attracted to one another because of the charges. And that's the important thing about ionic substances. Yes, they're split. They have donated things, but because they're still attracted with charges either to each other or when you mix them with water's polarity, its negative or positive aspects. That's what makes ionic compounds so important, and their ions important. So ions they've already left with the electron or lost their electron. And now we are hanging out in water instead. We're chill. We took the Barbie and we hung out with people we were more interested in. So that's the idea with ionic bonds. Ionization is whenever you have turned into ions because you went into the water and hung out with other other charges. All right, hydrogen bonds. Okay, so this is, I'm the oxygen, I'm nasty, I'm hogging the Barbie, and my sister's not getting as much, but we are still playing together. We're still sharing. We've still got our covalent bond going on. Think of couple, they're together, there's two sharing. Yeah, they're united. So we're united still, we're still playing together. So I'm not that nasty. And I've got the negative charge and she's got the positive charge because I'm just hogging the negative Barbie ion or negative Barbie electron, and she's not getting as much of it. All right, so because she's not getting as much of it, she has that positive charge. She's a hydrogen that is attached to something else covalently, me, the oxygen. And because I'm hogging her electron, she has a positive charge. That's the definitive of what starts the hydrogen bond. You have to have the hydrogen in it. It's not a hydrogen bond if there's not a hydrogen there. It's also not a hydrogen bond if your hydrogen didn't have a positive polarity to it. So because it has the thing hogged away, because I'm hogging her Barbie, and she has a positive charge, even though she's still connected to me, Now she can create hydrogen bonds with other negatively charged things out there. And when she does, that's the hydrogen bond. No electron exchange with that. It's just an association based on attraction. And one of those things that are attracted has to be a hydrogen. And hydrogens, when they have a charge, it's always going to be positive. Okay. So this kind of just goes through the wording, explaining it in more like actual professional terms. So one way that I could say you can remember that people share COVID, just like electrons are shared in covalent bonds. Or you can think of couples or co-parenting, you're working together, whatever it is that helps you remember what covalent means, okay? Valence literally has to do with your outer electrons, in case you forget what that means. So your outer electrons, co means you're sharing electrons. It literally says it in the word, but okay. So hydrogen bonds are when hydrogens say hi hydrogen to other atoms, okay? They're not hooking up with the other atoms. They're just being attracted to the other atoms. Okay. All right, this is the last one, by the way. The bond between magnesium and sulfate in magnesium sulfate allows it to dissociate into Mg plus and SO4 minus. Basically, we're talking about every twos, but let's not get into the semantics here, please. But they have the charges. That's the important thing, right? The Mg is going to have the plus charge. The SO4 is going to have a minus charge, and they're dissociated, and they're going to dissolve in water. So what kind of bond exists between the magnesium and the sulfate? Well, it's not covalent because they're not sharing, because we've actually got them separated with charges. They're not just polar, they're actually separate and dissociated in the water. We know that. It does. There's not hydrogen bonds because there ain't no hydrogen even here in this. And then it's not van der Waals because we didn't talk about that. And then it's not ionic. And that's okay. Like if you're like, oh, van der Waals, we didn't talk about that in this class, but we did talk about it in, Dr. Tim's class. And I don't know if you guys did or not, and I don't honestly care. But we didn't talk about it in my class. I'm not going to test you on it. So no. All right. And then ionic bonds, we did talk about them. We're going to completely donate electrons or lose electrons and then dissociate when we have other charged hydrogen and oxygen in water around us. All right, DNA exists as a double helix. It's called double because there are two strands held together exclusively by hydrogen bonds. Y'all tell me I didn't talk about this in class. If any of those given bonds, if any one of those bonds, sorry, I can't read right now, in any of those hydrogen bonds, right? What type of electron interaction is occurring? So if we had that ladder and the A is meeting up with the T, they are not. I already told you they're in hydrogen bonds, right? They're attracted by hydrogen bonds. So they are not doing what? Exchanging electrons. Only covalent and ionic have something to do with the electrons between the atoms. Hydrogen's just saying hi. Remember that. So we don't have electron sharing. We don't have electron donation. We have polar attractions, no electron transfers. Now that's making sense, right? Our hydrogen had the positive because the oxygen was attached to its hogging its electron. Same thing's happening on the other side, but it's the oxygen now talking to the hydrogen. The negative is talking to the positive. That polar attraction is what dictates the hydrogen bond, but one of them has to be a hydrogen. D says negative electrons become positive. Now we're just talking nonsense anymore. That's definitely not what's going on unless you have some sort of weird radioactivity or something. So we're not doing that. Right. All right, that is it. Thank you for attending today, those of you who did. I know it was definitely longer than I meant for it to be, but I hope that you guys got something out of it. I hope that you learned something out of it. And I hope that you guys are coming back to revisit this, are going to get something out of it as well, moving forward. So these are the topics that people miss the most. So if you understand these and you're good on these, either because of this or because you're going to spend more time on them because I told you they're the worst ones, then, now is after you do that, the time is to focus on the other things, right? Make sure you still get your chops on those. Yeah, my pleasure to help you guys out with this. If you guys do have any questions, let's just give a couple of minutes. You guys are welcome to filter out if you want to. I'm just going to wait to see if anybody has anything to say. If you don't, then I'm just gonna sign off and I will post this video in that discussion area. And the slides are also in the discussion for the weekly reviews. Yes. You guys are welcome. I'm glad it helped. Even if it just helps one person, you know, and that is like the reason. Try to get my tablet to work in the future so that I can write with the tablet in class as well as over Zoom. Let's see what happens. Just close it.
Recording in progress.
Just so I can hear myself. I'll mute this. Not smart enough. Except actually, y'all just sitting here while we're on the freaking iPad. I'm trying to see if I can get it to work with the Zoom. Let's see. Yeah, no one can see it. Oh, it's like writing a word.
Recording in progress.
Ridiculous. Okay. This isn't closing. Okay. So that. This is so much better. Okay. I figured it out. I had to be on here twice apparently, but this is gonna be a lot better. Those of you guys on Monday, Wednesday, I have the tablet, but Tuesday, Thursday, I don't. So this will help. All righty. I'll go ahead and head out. All right. I'll see you guys Monday, Tuesday. I gotta go share some activity.